Have you ever wondered how scientists can study the tiny powerhouses of our cells, like mitochondria or the cellular brain, the nucleus, all by themselves? It seems impossible, right? Like trying to separate the ingredients of a cake after it's been baked. But in biology, there's a clever technique for just that, and it's called cell fractionation. Today, we're going to dive deep into this process and see how it unlocks the secrets hidden inside every cell. Stay tuned, because this is where biology gets really cool. Cell fractionation is a fundamental method used in cell biology and biochemistry to separate different parts of a cell, known as organelles, so they can be studied individually. Think of it as a microscopic sorting facility. The whole point is to isolate specific components, like the nucleus, mitochondria, or ribosomes, from the rest of the cell. This is crucial because to understand what a single part does, you need to study it in isolation, away from the hustle and bustle of the entire cell. This process relies on two key physical properties, size and density. By separating organelles based on these differences, scientists can get a pure sample of, say, just mitochondria, and then analyze its structure and function in detail, often using powerful tools like the electron microscope. The entire process can be broken down into two major stages: homogenization and centrifugation. Let's start with the first one. The first step is homogenization, which literally means to make uniform. The goal here is to gently break open the cell membranes to release all the organelles and cellular contents into a solution, creating a smooth, soupy mixture called a homogenate. Imagine putting a bunch of fruits into a blender to make a smoothie. It's a similar idea, but way more delicate. Scientists use special equipment like a mechanical homogenizer, which is like a tiny, precise blender, or even high-frequency sound waves in a process called sonication. The cells are suspended in a special buffer solution. This isn't just any liquid. It's carefully prepared to mimic the cell's natural environment. It's kept cold to prevent enzymes from digesting the organelles, has a specific pH to maintain their structure, and contains sugars to prevent them from bursting due to water intake. For plant cells, which have a tough cell wall, an extra step is needed. Enzymes like pectinase and cellulase are often added to digest the wall and make the cells easier to break open. Once we have this uniform suspension of cellular parts, we're ready for the main sorting event, centrifugation. Now for the second and most exciting part, centrifugation. This is where the actual separation happens. The cell homogenate is placed in test tubes and spun at incredibly high speeds in a machine called a centrifuge. This spinning creates a powerful force, called centrifugal force, which is much stronger than gravity. This force pulls the components of the mixture outwards and downwards. Just like when you're on a fast-spinning merry-go-round and you feel pushed to the outside, the same principle applies here, but on a much smaller and faster scale. The key is that heavier, denser particles experience this force more strongly and move to the bottom of the tube faster than lighter, less dense ones. This allows us to separate the organelles based on their size and density. There are a couple of ways to do this, but the most common method used for initial separation is called differential centrifugation. Let's break down differential centrifugation. The differential part means we do it in steps, with each step separating out different components. We start by spinning the homogenate at a relatively low speed. The largest and densest organelles, like the nuclei, can't stay suspended and are forced to the bottom of the tube, forming a solid clump called a pellet. The rest of the liquid, which still contains all the smaller organelles, is called the supernatant. We then carefully pour off this supernatant into a new tube and leave the pellet of nuclei behind. Now, we take the new tube with the supernatant and spin it again, but this time at a much higher speed. This increased force is enough to pull down the next heaviest organelles, typically the mitochondria and, in plant cells, chloroplasts. They form a new pellet at the bottom. Again, we pour off the supernatant into another tube. We can repeat this process several times, each time increasing the spinning speed. After mitochondria, we can pellet parts of the cell membranes, and finally, at very high speeds for a long time, we can even get the tiniest particles, the ribosomes, to form a pellet. Each pellet gives us a progressively smaller and less dense fraction of the cell's original contents. So, through this step-by-step process of differential centrifugation, scientists can effectively build a library of isolated cell parts. They'll have one tube with just nuclei, another with mitochondria, a third with fragments of membranes, and so on. These purified samples are now ready for experimentation. Scientists can study the biochemical reactions that happen only in the mitochondria, analyze the proteins found in the nucleus, or figure out the exact structure of a ribosome. It's this ability to deconstruct the cell and study its components individually that has allowed us to understand so much about how life works at the molecular level. From discovering the role of mitochondria in energy production to understanding how DNA is organized in the nucleus, cell fractionation has been at the heart of countless biological breakthroughs. And there you have it, the elegant and powerful technique of cell fractionation. It's a perfect example of how physics and biology work together to help us explore a world that's invisible to the naked eye. By breaking cells apart and sorting their contents, we can piece together the intricate puzzle of life, one organelle at a time. I hope you found this journey into the cell as fascinating as I do. If this video helped you understand this complex topic, please give it a thumbs up, share it with anyone who might be interested, and don't forget to subscribe for more deep dives into the amazing world of biology. Thanks for watching, and I'll see you in the next video.